Welcome back to another video tutorial. So in which we'll be solving second year DMLT Microbiology October-November 2023 question paper. Okay, let's get started. The first main is short notes, and the first question is write the principal, uses and sterilization control of hot air oven. So what is this hot air oven now? So it is a laboratory equipment used for sterilizing materials using dry heat. It ensures destruction of microorganisms by circulating hot air at controlled temperatures. Yes, so it is a main equipment which we use it in the laboratory. So it is used for sterilizing materials. Okay, and the main principle is dry heat. Okay, so throughout the equipment, dry hot air is circulated, so which will cause oxidation in the cellular components of the microorganisms, and it also denatures the proteins in the microorganisms. As a result, the microorganisms would die. So denaturation means here it is changing the structure, or it is destroying the secondary and tertiary structure of the proteins. So as a result, proteins are losing its functions. So when the proteins inside the microorganisms are losing its functions, so microorganisms are dying here. Okay, so it usually maintains a constant temperature, so which is 160 to 180 degrees Celsius. Okay, now uses sterilization of glasswares like test tubes, flask, petri dishes, pipettes, and metal instruments like scalpels, forceps, and it is also used for the items where we cannot use moist heat. Okay, like for example oils, fats and powders. So if we use moist heat sterilization, they would get damaged. Okay, such items we can sterilize in hot air oven. And then syringes and other heat stable items. And what are the sterilization conditions is 160 degrees Celsius for 2 hours, so 170 degrees Celsius for 1 hour depending on the load. And sterilization control, so there is physical control, so where temperature and time monitoring using thermometers or automatic timers are used, and these are inbuilt. Okay, usually they will be present inside the hot air oven, and so they will monitor the temperature. Okay, and automatic timers also will be present. And apart from that, second is chemical indicators. Chemical indicators like Brown's tubes, dry heat indicator tapes are available, so which we can use it. So what is this Brown's tube? No, Brown's tube is the small sealed glass tube which contains a heat sensitive chemical solution. Okay, so they we can insert them in the hot air oven. Okay, so and so they will show color change, so when the correct temperature and time has been reached. Okay, so in the similar way we also have dry heat indicator strips. So these are adhesive tapes, so they are coated with sensitive chemical dyes, so they also will change color when exposed to the required sterilization temperature. Okay, so these are the indicators to know whether a particular temperature is reached and whether the sterilization has happened effectively or what. So the third indicator is biological indicators, so where we use spores of Bacillus subtilis. So the destruction of the spores confirms that the sterilization is done effectively. Okay, and the second question is draw a neat label diagram of compound microscope. So this is a diagram of compound microscope with labeling. And then so next third question is Zeal-Nielsen staining, okay, which is also known as acid fast staining. So this is a differential staining technique. Especially we use this to identify acid fast organisms like Mycobacterium tuberculosis, Nocardia, etc. Okay, and so why do we do this special staining technique is because these organisms, no, they have a waxy mycolic acid in their cell wall. So because of this, no, so they are resistant to ordinary stains, but when they are stained it is difficult to decolorize as well. Okay, so that's why we do this special staining technique, so in which we use a primary stain called carbamazepine and acid alcohol as the decolorizer, and counter stain which is methylene blue. Okay, so coming to the procedure, prepare and fix the smear on the slide. Flood the carbofucsin and heat gently for 5 minutes. Yes, after preparing a slide, air drying, heat fixing and all those things, we will flood it with carbofucsin. So, but immediately it will not enter the cell. So we need to heat them gently for 5 minutes. When we heat, what's happening? We're forcefully making the stain enter inside the cell. Okay. So once the acid fast bacteria takes or absorbs this carbofucsin, we cannot decolorize them very easily. Okay, so now forcefully after introducing, we will decolorize with acid alcohol. At this particular step, what happens is, if it is acid fast bacteria, so it will not get decolorized. Only if it is non-acid fast bacteria, so it will get decolorized. And so when we counter stain with methylene blue for 1 to 2 minutes, only the non-acid fast bacteria will take up the stain and they appear blue in color. So whereas the acid fast bacteria, they always appear red in color because they will not get decolorized itself. Okay, so then we will wash, dry and absorb under oil immersion. Okay, now coming to the results of it. So acid fast bacteria will appear red or pink in color and non-acid fast bacteria will be blue in color. Okay, so coming to the uses of it, so they are used in the diagnosis of tuberculosis, leprosy and other mycobacterial infections, and so they are also used in the detection of nocardia species in clinical samples. And next, question number 4 is active immunity. So yes, active immunity is a type of immunity in which the protection is produced by the body's own immune system. So our immune system is the one which is fighting against the pathogens. Okay, so in case we are encountered with any infection, or forcefully we are introducing any microorganism through vaccination. Okay, so our body fights against it. They form antibodies which are very much specific and also form memory cells. So this memory cells will have a long term memory. Okay, so and the next time when the same infection or when the same microorganism is entering you, so they immediately show response. So that is active immunity. So it's of two types. First is natural active immunity. So it develops after a natural infection. Naturally you get some infection, your body fights against it, they produce antibodies and they produce memory cells. That is natural active. Okay, now next is artificial active immunity. So as the name tells here, artificial. So in this method, we are artificially introducing the microbes into the body through vaccination. Okay, so we know that vaccines do contain killed or attenuated microbes. When we introduce them into the body, our body fights against it. Okay, so that is artificial active immunity. So the mechanism here is when the antigen enters the body, B lymphocytes and T lymphocytes will be activated, and B lymphocytes will produce antibodies. So these antibodies are very much specific. They go target and kill the antigens, and T lymphocytes will produce memory cells, and these memory cells are long lasting. Okay, so that is next time when the same microbe or when the same infection occurs in you, so they immediately go and they fight against the antigen. Okay, so and the main features of this is they are lifelong, they are long lasting, and the antibodies are highly specific. Okay, example is immunity after chickenpox infection. We all know that a human gets chickenpox once in their life, after which they do not get, is because of this active immunity. And then immunity developed after receiving hepatitis B vaccine. And the next question is delayed hypersensitivity. So this is type 4 hypersensitivity. So why is it called delayed? Is because so they occur a day or after that. So it takes time, okay, to show the immune response. That's why we call it as delayed hypersensitivity, and it is cell mediated. Okay, cell mediated. Why? Because in this type of hypersensitivity, antibodies will not be formed. Okay, hence we call this as cell mediated immunity or immune response. But we can see T lymphocytes and macrophages in this type of hypersensitivity. Okay, So now coming to the mechanism, there are two phases involved here. First is sensitization phase and second one is effector phase. So sensitization phase is the activation phase. Okay, so when the antigen is exposed for the first time, so T helper cells will be activated. Okay, so whereas in the effector phase, when the same antigen is re-exposed, so this sensitized or activated T helper cells, no, they release cytokines. Okay, so and that recruit the macrophages and they cause inflammation and tissue damage. Okay, examples for this is tuberculin test or Montauk's test, which is done for TB. So initially, what is this Montauk's test? It's a test in which the tuberculin purified protein derivative, that is PPD, a sterile solution which is made from the protein of tuberculosis causing bacteria, is injected just under the skin on the inner forearm. Okay, and the resulting skin reaction is measured after 48 to 72 hours to determine the test result. So now this test is showing type 4 hypersensitivity, right? And the second example is contact dermatitis, which is the reaction of poison ivy or nickel. And then next is leprosy granulomas. So this is actually the microscopic clusters of immune cells, so primarily macrophages and lymphocytes that form in response to Mycobacterium leprae infection, which is a key feature of the disease. So all these are examples for type 4 hypersensitivity. The main features is, as I told, it happens, the reaction develops after 24 to 72 hours, or sometimes it might even take more time. So hence we call it as delayed hypersensitivity. It is cell mediated. Why? Because there is no antibodies formed here, and it causes local inflammation, redness, swelling, tissue damage, or sometimes even more. And then the significance is, as we studied, so it is used as a diagnostic tool in tuberculosis and leprosy, and it is important in chronic infections and transplant rejection. Okay, so next question is fluorescent microscope. So yes, this fluorescent microscope is a light microscope, so which uses fluorescent dyes or naturally fluorescent substances. And so this fluorescent microscope will produce a green color cells, okay, green color fluorescent cells under the dark background. And usually we use this for detecting the autoimmune antibodies or antigens. Okay, so which will produce a very good contrast and it will show a specific structures. So it mainly works on the principle of fluorescence. Okay, so what is fluorescence now? So when any substance, no, so they absorb shorter wavelength of light and they emit longer wavelength of light, so which is detected to form an image, so that is called as fluorescence. So in this fluorescent microscope, what we do, so the specimen which is to be observed, no, we tag the specimen with a fluorescent dye or naturally fluorescent substances. Okay, and then we visualize them under the microscope. So now what are the components which we use here is, first is the light source. So light source is usually UV or blue light. Okay, and the excitation filter, so excitation filter will allow only a specific wavelength to pass. As you can see here, there are different colors, but it is allowing only one color, blue light, to pass through. Okay, and then there is a dichroic mirror, as the name tells, dichroic. So what does it do? It will allow one wavelength of light to pass through. Okay, and it will also send another wavelength to the ocular. Okay, so one wavelength is moving, another wavelength is moving out, so that's why we call it as dichroic. And then, so yes, now what is happening here is, blue light is passing through the specimen. Since there is fluorescent tagged material here, it is producing the longer wavelength light, that is green light. Okay, so here blue light is shorter wavelength, green light is longer wavelength. It is taking the shorter wavelength light and it is emitting the longer wavelength light. Okay, so that moves through the emission filter, which is also called as barrier filter. So this blocks the excitation light and allows only the emitted fluorescence to reach the eye. So and the objective lens of the eyepiece will magnify the fluorescent image. Okay. Okay. So now the uses of fluorescent microscope is, they're used in detection of microorganisms, They're used in identification of antigens and antibodies using fluorescent tagging. They're used in studying cell structures, organelles, and proteins in research. And the advantages or significance is high sensitivity and contrast, and can detect specific molecules in complex samples, and it is useful in diagnostics and biomedical research. The next question is difference between prokaryotic and eukaryotic cell. So here is the difference. So first difference is in nucleus, and this is a main difference. Yes, so in prokaryotic cell there is no true nucleus. DNA is in nucleoid. That means yes, DNA is lying in the cytoplasm. Okay, they do not have a membrane bound nucleus present here, hence we call it as nucleoid. So whereas in eukaryotic cell true nucleus is present, so which is enclosed by a nuclear membrane. Okay, and in prokaryotic cell membrane bound organelles are absent. In eukaryotic cell they are present, like mitochondria, endoplasmic reticulum. So all these are membrane bound. Okay, and coming to the cell size, prokaryotic cells will be smaller, eukaryotic cell larger, and DNA structure, they will be circular and naked DNA. So they will have linear DNA associated with histone proteins. Example is bacteria, example is plants, animals, etc. Okay, and then next question is properties of antigens. So what is antigen now? So antigen is a substance that can stimulate the immune system. So that means when any foreign substance enters your body, if your body has a capacity to fight, so it is showing immune response, it is producing antibodies, so such substances are called as antigens. So coming to the properties of it, first is immunogenicity, as I told. So when the antigen enters, your body has to show the immune response, they have to produce the antibodies. Okay, and then secondly, antigenicity. So this antigens, no, they should specifically react with the products of immune response. Now for example, if the antibodies are produced, if the T cell receptors are produced, so all that components or products, no, so they should be specifically able to react. And then next is specificity. Each antigen induces antibodies specific to its unique structure. Yes. Okay, that means now each antigen, no, they trigger the production of the antibodies. Okay, and antibodies are so specific that they can recognize and bind only to that specific antigen. Okay, and the next property is foreignness. So that means antigen must be recognized as non-self. It is foreign. Okay, it's from outside. And then molecular size and complexity. Usually larger molecules like proteins, polysaccharides, glycoproteins with complex structures, no, they will be more antigenic. So examples is bacterial cell wall components like components, viral proteins, toxins, pollen, and transplanted tissues. Okay, so the bigger they are, that much antigenic they will be. Okay, and the next question is contributions of Robert Koch. Okay, so yes, Robert Koch, no, he is a German microbiologist. He is known as father of modern bacteriology, and he is very well known for germ theory of disease. Okay, so he has given postulates which is famous. So what is this postulates given for? It's mainly a criteria to prove that a specific microbe causes a specific disease. That means, so this microbe itself is causing that particular disease. Okay, so to know that, you have to follow the Koch postulates. Okay, and the second contribution is discovery of pathogens. So he identified the causative agents of tuberculosis, that is the organism Mycobacterium tuberculosis, and cholera, which is Vibrio cholerae. And he also identified pure culture techniques, that is he developed methods to isolate and grow pure bacterial cultures on solid media. And then some staining techniques, like he introduced techniques to visualize bacteria using specific stains. And then he was awarded Nobel Prize in Physiology and Medicine in 1905 for his work on tuberculosis. Okay, so yes, his work laid the foundation of microbiology and for infectious diseases research and modern medicine. And the next 10th question is indole test. So yes, this indole test is a biochemical test which is used to determine the ability of bacteria to produce indole from amino acid tryptophan using the enzyme tryptophanase. So what is the basic idea is that some bacteria will contain this enzyme which is called as tryptophanase. So when this enzyme is present in them, no, they can break, okay, tryptophan, and they can produce indole. So to detect whether the organism can produce indole or what, we do this particular test. See, the detailed principle is here. See, bacteria with tryptophanase, when the bacteria contains this enzyme called as tryptophanase, it breaks down tryptophan to indole, pyruvate and ammonia. Okay, so indole, no, this reacts with Kovacs reagent. So when you add Kovacs reagent, it reacts with this Kovacs reagent and it forms the red color ring. Okay, so the procedure is you need to inoculate the test bacterium in tryptophan broth. You need to incubate them for 24 to 48 hours, and then you will add Kovacs reagent. So when you add the Kovacs reagent, what happens? If indole is present, if it is broken down, so indole will react with the Kovacs reagent, they form the red colored ring. So you have to observe for the color change. In the results, positive, if it is positive, red color ring will be there at the top, indole is produced, and if it is negative, no red ring, so indole is not produced. Uses, so they are mainly used for identification of Enterobacteriaceae members and differentiating E. coli from Enterobacter. And yes, it's a simple and rapid diagnostic tool in microbiology for bacterial identification. Okay. Yes. Okay. Second main short answers which carries 3 marks. 11th one, draw a neat label diagram of IgG. So this is the structure of IgG, immunoglobulin G, and so this is a L chain, FAB region, Fab region, antigen binding site, and FC region, and these are the labelings. The next 12th one is name 3 agglutination tests. So first is Vidal test for typhoid fever, VDRL for syphilis, latex agglutination test for detecting bacterial or viral antigens. And then 14th one, name 3 materials sterilized in autoclave, culture media like broths, agar plates, and glasswares like test tubes, flask, pipettes, surgical instruments, or heat stable medical equipments. Next is cold sterilization. So what is cold sterilization? Is a process of sterilizing heat sensitive materials, so through which we cannot apply heat, and we use chemical agents here for sterilization like ethyl alcohol, formalin, glutaraldehyde, hydrogen peroxide, etc. And then mainly we use this for plastic wares, surgical instruments, catheters, and other heat liable items. Name 3 spore bearing bacteria: Bacillus anthracis, Clostridium tetani, and Clostridium botulinum. Then 17th one, mention different types of microscopes: light microscope, electron microscope, and dark field microscope. And then 18th one, name 3 inoculation methods of culture. So it is streak plate method, pore plate method, spread plate method. Name 3 routes of transmission of HIV. It's through sexual contact, blood transfusion or sharing needles, mother to child, which is called as vertical transmission. Then 20th one, name 3 chemical disinfectants. It's phenol, alcohol, and glutaraldehyde. 21st, name 2 differential stains. So first is Gram stain, second is Zeal-Nielsen stain or acid-fast stain. Okay, so 22nd one is standard precautions to prevent hospital acquired infection. So first is hand hygiene, which is important. So wash hands with soap and water or use alcohol based sanitizer before and after patient contact. Use of personal protective equipment, that is PPE, like gloves, masks, gowns, and eye protection when exposed to blood or body fluids if possible. And then safe injection practices. Use sterile needles and syringes and avoid reuse. So 23rd is hand hygiene. Yes. Hand hygiene is a practice of cleaning hands. So it's mainly to remove dirt, microorganisms, and to prevent the spread of infections. So hand washing has to be done with soap and water for at least 20 to 40 seconds. And hand rubbing has to be done by using alcohol based hand sanitizers when hands are not visibly soiled. And then prevent hospital acquired infections and transmission of pathogens between patients and healthcare workers. It prevents. So this is very important to prevent. So this infections and transmission. And then 24th, function of bacterial capsule. So yes, bacterial capsule is outermost layer of bacteria. So it shields the bacteria from phagocytosis by host immune cells. Phagocytosis is nothing but engulfing. Okay, so when the immune cell comes, it swallows inside, we call it as phagocytosis. So capsule is the one which protects against that, and then it prevents desiccation, helps retain moisture and survive in harsh conditions also. And then adherence aids in attachment to surfaces and formation of biofilms, enhancing colonization. It helps in attaching itself. And then name three motile bacteria. Motile bacteria is a bacteria which shows movement, which has flagella in it. So first one is Escherichia coli, second is Salmonella species, and third is Proteus mirabilis. And then name three anaerobic culture media. So that is which do not contain oxygen. So thioglycolate broth medium, anaerobic jar, that is gas pack method, and then anaerobic chamber glove box. 27th, what are the ingredients of nutrient agar? Peptone 5 grams per liter, beef extract 3 grams per liter, sodium chloride 5 grams per liter, agar 15 grams per liter, and distilled water will be 1 liter. pH is adjusted to 7 plus or minus 0.2. Biomedical waste segregation. Segregation means it is separation of the biomedical waste. Okay, and so these are the color of the containers which we use for it. In the yellow bag or container, we have to put infectious waste, human tissues, and sharps after treatment. And then in the red bag or container, contaminated plastic, tubings, and disposable items. And in the blue or white puncture-proof containers, sharps like needles, syringes, or any other sharp objects. Okay, and then 29th is COVID-19 virus. So yes, COVID-19, as we all know, it is caused by SARS-CoV-2, a novel coronavirus that primarily affects the respiratory system, and the transmission is by respiratory droplets and close contact with infected individuals. So symptoms will be fever, cough, fatigue, loss of taste or smell, and shortness of breath. So severe cases may lead to pneumonia, acute respiratory distress, or even death. So prevention is by vaccination, masks, hand hygiene, social distancing, and ventilation. So then the last question is name three live vaccines. So where the organism is injected or the microbes are injected live. So first is measles, mumps and rubella MMR vaccine. The second one is oral polio vaccine OPV, and next is BCG vaccine which is given for tuberculosis.